So we're going to continue on now with host defenses, and we're going to be looking at the third line of defense. This is what we tend to think of as immunity. We don't think of the other first and second line of defenses. We don't think of those when we consider immunity. When we think of our immune system, we think of the third line of defense. This is going to be our acquired immunity. So these are things that we pick up as we go. This is not something we're born with, okay? It's very specific, targeted to one particular antigen, okay? One particular invader, all right? So very specific here, okay? And so third line of defense. When our first line and second line barriers do not stop the infectious agent, then our third line of defense, our immunity here, is going to kick in. So again, this is what we normally think of as immunity. We are not born with this. We acquire it in our life as we live our life after we are exposed to various pathogens. This is where our antibodies generally lie, okay? Our B cells and our T cells. Acquired or adaptive immunity is the product of B and T lymphocytes. These are very specific to one specific antigen or immunogen, okay? An infant is born with the potential to react to a vast, innumerable amount of substances. So we have what we call a great immunocompetence. That means the ability to react with basically any foreign substance, any pathogen or antigen that we come into contact with. We have a match. Now, it's one single match, okay? And so until we come into contact with it, these aren't utilized. They're just sitting there, okay? And so it's actually just amazing to me because these are all based on shape of those epitopes on the antigens, okay? It's amazing to me that we potentially have millions of these, millions of different combinations that will match any other piece that comes in. It's like a puzzle piece. So if you had a puzzle made of two pieces, you've got this foreign invader coming in that you've never seen. Your body is prepared with one of those pieces, okay, somewhere. Now, in order to defend it off and have what we call a memory response or become immune to something, we have to encounter it and make copies of those pieces so that we can trigger the reaction much faster. But it's fascinating to me that our body is already equipped. Now, and don't confuse this because we do talk about it being acquired immunity. You're not immune to something until you encounter it, but you have the predecessor or precursor molecules, so to speak, ready there waiting to come into contact with something. So antigens, and we've talked about them some, but these are surface molecules made of proteins, polysaccharides, the sugars. These are sensed and identified by the immune system. And so these antigens are kind of like a name tag. So if they provoke an immune response, then we call it an immunogen. All right. So these are not just pathogens or microbes. These can also be pollen, environmental chemicals, other things like that. They don't have to be a microbe. Our own antigens usually do not evoke a response from our own immune system. We call that recognition of self. If it's supposed to be there, we recognize that little antigen or name tag as self. If it's not supposed to be there, we recognize that and label it as non-self, okay? And so a lymphocyte is so specific that it only responds to a very, very small portion of the antigen called the epitope. Now, I tend to kind of throw those words around the same. But so here you can see there's like that triangle shaped antigen, okay? So if this is the triangle shaped antigen, this is the epitope here, okay? So it's just a little tiny portion there, but it's got to match, all right? Remember, structure or shape determines function, and the shape has to match. So I've got to have something shaped like this to match with that. Okay, it's got to have this shape where it can fit together. And of course, that's a very, very simplified model there, okay? Antigens, we've already said, are highly individual and they stimulate specific immunity. We have 6 characteristics that make the immune system unique: specificity, diversity, inducibility, clonality, tolerance, and memory. Two of these are specificity and memory, okay? Specificity, the antibodies are produced during an infection of, say, the chickenpox virus, will function against that virus, but not against the measles virus. That is specificity, okay? Memory is that rapid response to or mobilization of lymphocytes to recognize that invader more quickly and have a much bigger response the second time around. Okay, and so once I'm exposed to that chickenpox virus, I will develop an immunity. I will develop a memory, sorry. And so that is how I will have immunity, because I will make many copies of the particular antibodies responses that we have made to this. And so when it comes back, it has no chance. So how do these progress along? How do these stages progress along once we have come into contact with something, okay? So these are the stages of immunologic development and interaction. So lymphocytes work close together in association with phagocytes. They work closely together. So these here are the stages we're going to look at. Stage 1, lymphocytes. They're going to develop, and we're going to have clonal deletion and differentiation. Deletion basically just means that they're going to get rid of ones that have prematurely ID'd themselves as self before they have completed maturation, okay? So this actually helps prevent autoimmune disorders. All right, so when these cells are very young and they have not been sent out yet, they have to go through a process of maturation and then put on their little name tag. Well, they put their name tag on too early, so we have to get rid of them. Okay, differentiation occurs in the bone marrow for antibodies and the thymus in the bone marrow for B cells and in the thymus for T cells. Okay. Then we're going to have presentation of antigen. We have antigen presenting cells. We'll have clonal selection. We're going to go through all of these. Challenge of B and T lymphocytes by antigens. And then we're going to have the T lymphocyte and B lymphocyte responses. All lymphocytes are going to come from the same basic stem cell type. Okay, and then they're going to develop into B cells, all right, or T cells, okay? And so once they mature in the bone marrow, after they undergo clonal deletion, they will migrate and become a B cell and a T cell, okay? And so here we're going to go to the bone marrow, and then they can move on to the lymph node or the spleen. Here it's going to be, they're going to finish maturing in the thymus, and then they will go on to the lymph node or the spleen as well. So it's kind of like they have a two-stage maturation process. Okay, they're going to mature a little bit in the bone marrow or the thymus. Okay, so bone marrow for B cells and thymus for T cells. All right, so both cell types migrate to separate areas of lymphoid organs. All right, B and T cells constantly recirculate through the circulatory system and the lymphatic system, and they migrate in and out of these lymphoid organs. All right. Development of your B and T lymphocytes, specificity, and the migration to these organs is going to occur. All right. Lymphocytes are really important to immune responsiveness. They're going to undergo development that actually begins all the way back in the embryonic yolk sac and shifts to the liver and bone marrow. Although all lymphocytes arise from the same basic stem cell type, at some point in development, they're going to split off into those two types. The final maturation of B cells, like we said, in bone marrow, T cells in the thymus, both are going to migrate to those lymphoid organs, and then they're going to recirculate, as I already said, okay? Antigen processing by dendritic cells and presentation to lymphocytes is going to be assistive to B cells by T cells. We have what we call T helper cells, okay? And so next we're going to have antigens that make contact with some markers. The antigen can be processed by a phagocytic cell, and in this case over here to the left, by a dendritic cell here. okay? And so the dendritic cell then will digest and display that antigen on its outside surface. It's basically like a little help wanted sign, okay? It's going to display that to the helper T cell, okay? This textbook says T helper cell, same thing, all right? And so it's going to display it to the T cell, and the T cell can go and send it to this activated cytotoxic T cell. It can become a specialized effector cell, different things like that, okay? Also can go over here and stimulate the B cells, okay? The helper T cells can. And so we have gamma T cells as well that can be activated. You can see over here, these can go on also to some cytotoxic T cells, which are basically killer cells. The B cell can also be differentiated down here into plasma cells, which will make antibodies. All right, so when non-self cells enter tissues, phagocytes are going to come on the scene, ingest the pathogen, and produce inflammation. Dendrites ingest that antigen and go to those nearest lymphoid or to the nearest lymphoid organ and find those helper T cells to present it to, and then that's carried on to those B lymphocytes. Okay, so pathogens cross that first line of defense. The resident phagocytes are going to rush in. The macrophages can ingest that pathogen and cause inflammation, and dendritic cells can ingest the antigen and go and tell the helper T cells, make a help wanted poster, and draw those B cells in to come and fight off, okay? Those T and B lymphocytes, technically. So what are these little name tags or surface markers, okay? All cells have a variety of different markers on their surfaces. They play an important role in activating different components, particularly for immunity, okay? So we're going to focus mainly on the major markers for lymphocytes and macrophages because those are what are important to the immune system. So some of the functions here for surface markers are detection, recognition, and even cell communication. They are what we call a major histocompatibility complex. This is a set of genes that codes for human cell markers or receptors. It's basically a little sign on the outside that's like a name, okay? This is also known as human leukocyte antigen or HLA. And so this is important to the immune system. It is also important in things like tissue transplants. We have other markers on the outside of a cell also that we call clusters of differentiation. You'll hear me talk about CD4 and CD8 later on. There are over 300 of these that have been named, and it's basically a naming scheme for some of these other surface markers. Many of these are involved in the immune response, but like I said, we will talk later on about CD4 and CD8, and you can see this is just a little kind of variety showing you the different structure and function, and so there you have different names, of course. Lymphocyte markers can also act as receptors. They have to accept or grab a hold of and grasp antigens. B cells have receptors that bind antigens. T cells have receptors that bind antigens that have been processed and complexed with those MHC, major histocompatibility complex molecules, on the presenting cell surface. The receptors have the capacity to respond to an infinite number of different antigens. So when they are presented or challenged with an antigen, then they come to contact with them. The B and T cells are going to proliferate, which means copy over and over and over again, and differentiate. So they're going to make clones of identical cells. And so some will go to be memory cells to ensure future reactiveness against that particular antigen. And then the responses that occur from this point on are going to differ a little bit from B cells and T cells. So we have many different types of T cells, but these are some really important ones here. And then we have B cells. So we have our three functional basic types of T cells. We have our helper T cells, which can activate those macrophages, assist B cell processes, and help activate cytotoxic T cells. Then we also have regulatory T cells, which control your T cell response, okay, and cytotoxic T cells, all right, which lead to destruction of foreign cells. T cells secrete cytokines in order to help destroy pathogens, but they don't produce antibodies, okay? Neutralization is the process where antibodies, I'm getting ahead there, okay, sorry. So, very important point there that T cells do not produce antibodies. When activated, B cells divide and give rise to plasma cells. Plasma cells then release or secrete antibodies, okay? So your antibodies are going to come from your plasma cells. B cells are going to divide and differentiate into plasma cells that produce antibodies and memory cells, all right? And so then those are going to go and attach to the antigen for which they are specific and neutralize them. They're going to mark them for destruction and neutralize them. Neutralization is the process where antibodies cover a virus or foreign molecule and basically disrupt their activity. They surround it. All right, so here we're looking at some different properties of B cells and T cells. This is important to know the differences, particularly B cells are going to mature in the bone marrow, T cells in the thymus, okay? The surface markers, B cells are going to have immunoglobulins, which are basically antibodies kind of attached to them. They're not free-floating. And then we have T cell receptors and those clusters of differentiation over here for T cells. In the blood, you will not find very many B cells, but you will find very many T cells. For antigen receptors, the B cell receptor is going to be the immunoglobulin. Let's see. Okay, antigen presentation. T cells require, B cells do not, although they are sometimes assisted by T helper cells. B cells produce plasma and memory cells. Okay, that's really important there too. Several types of activated T cells and memory cells over here for your T cells. Okay, general functions, these are important. Production of antibodies, again, very important. These are the ones that produce antibodies, and then that will inactivate, neutralize, and target antigens. T cells are going to be activated, act as those helper T cells. They can suppress or kill abnormal cells. They can be cytotoxic T cells, which go out and kill. They can mediate or help control hypersensitivity, and they can actually make cytokines. By the time B and T cells reach lymphoid tissues, each one is equipped to respond to a single unique antigen. Diversity, well, before I say that, it's estimated that each human being produces antibodies with the ability to match 10 trillion different foreign invaders. So we have 10 different trillion specificities. That diversity is created by a rearrangement of those genes, of those variable regions of antibody molecules because of things like alternative splicing, different RNA transcripts, and from the original gene. So it creates all these differences, okay? When those transcripts are translated, the protein will have a variable amino acid sequence and therefore a variable shape. So every possible recombination can occur, leading to a huge assortment of lymphocytes. So this illustration here, you've got the gene encoding for those variable regions, and they're kind of all shuffled up. And then here we have those different amino acid sequences in the finished product. This is the classic appearance of an antibody, the Y shape of an antibody. And we do have variations on this, but each of them has what we call a variable region, okay? And so this is where they're going to be different at the top here. Down at the bottom here is what we call the constant region. And so that's where they're all basically the same. All right, so here are some antibodies, okay, that also known as an immunoglobulin, Ig. All right, you'll see we're going to look at 5 different types of antibodies. These are large sugar proteins, so glycoprotein molecules, and they serve basically as antigen receptors on B cells, okay? You can also have freely floating in the blood system after they are secreted or removed from the B cell, then they're called antibodies there, okay? That's the main difference. Are they attached to the cell or have they been released from the cell? Not that big of a difference. So they have these antigen binding sites, which are pockets in the ends of these forks up here on the molecules that can be extremely variable, as we already said, and so that shape is to fit a wide range of different antigens. Okay, so those variable regions are extremely versatile and different from one clone to another. We have what we call light chains and heavy chains. Okay, this is the light chain here, and these are the heavy chains here. Okay, we have our constant region that we looked at, and then the variable regions up here. Okay, so while B cells and T cells do have a little bit different processes, their receptors do belong to the same family of proteins. They are similar to B cells because they do have that genetic modification. They do have variable and constant regions. These are going to be inserted into the membrane and have an antigen binding site. These are really, really small, and they're never really actually secreted, though, like antibodies are secreted.